Hi there, I'm Nate Nowak from Transgenetics, Vice President of Scientific Affairs. Thank you for joining us today for the first in a series of webinars from Transgenetics. Please look for announcements for these other webinars in the future. We hope you'll attend those also. We'll be recording today's presentation and sending out that link once the recording is posted. Questions can be submitted by using the question feature on the right-hand panel. We will answer questions at the end of the presentation, but please feel free to submit them at any time. If there are more questions than we have time for at the end, we'll put together answers for any that we don't get to and send those out as a follow-up. Following the presentation, I'll give you some information about Transgenetics' genetic monitoring service. Now, I'd like to introduce Dr. Fernando Benavides. Dr. Benavides receives his DVM and Ph.D. degrees from the University of Buenos Aires, Argentina, with Ph.D. training in the Pasteur Institute in France. He is a diplomat of the American College of Laboratory Animal Medicine since 2003. He is now professor at the Department of Epigenetics and Molecular Carcinogenesis and the director of the Laboratory Animal Genetic Services at MD Anderson Cancer Center. Dr. Benavides has over 50 peer-reviewed publications, 2 books, 5 book chapters, and several reviews on the subject of mouse genetics, mouse models of cancer, and genetic quality control of inbred strains.
Thank you, Nate, and thank you, Transgenetics, for the invitation, and for all out there, all those that are joining the webinar. So we're going to be talking about this important issue about genetic background. Let's start moving. What are the concerns that we have about the quality of the mice that we are using? So let me explain a little bit why this is so important. I'm going to present a few slides in the beginning very shortly, only to point out that these are important, but I'm not going to explain in detail. For example, nomenclature is something that is really important. Very few people is really taking this seriously, and that's many times a big issue. Many publications we can see very bad nomenclature, and nomenclature is very important because it's not that we are picky and that we want to follow this standard nomenclature, but it's because nomenclature is information. I mean, we need to be sure that we use the correct nomenclature, and also that we understand the nomenclature, how nomenclature works. It's like almost knowing the language, because it's not that easy, but we need to take time. I mean, it's important to take the time to read how this nomenclature is working. We have everything online on this link. You can find all the information. It's been revised lately in 2016, and it's the same for mouse and rat strains. So genetic monitoring, it's been a term that was used for decades already. It was originally thought only to be applied to cases of genetic contamination, which was something that happened frequently, as I am showing here on this publication from the 80s, the 90s, some from early 2000. I mean, mice can be accidentally crossed, and if they are from different inbred strains, of course, the result is that we have contaminated animals. So that originally the term genetic monitoring was used only to control or validate or authenticate the purity of different inbred strains. That's the way it was used in different publications at the time. Now we can extend a little bit this genetic monitoring term to the background characterization and other important points I'm going to be showing you in details. Of course, only 1 or 2 slides here to talk about how to prevent the genetic contamination. And basically, what many places are doing now is embryo freezing, which is really important because in this way we prevent, of course, not only genetic contamination, but we also prevent this genetic drift that I will explain later, how mutations accumulate with time, and the different animals on the same inbred strain could lead to different substrains with time. The use of ventilated racks and isolators, of course, is helping also, but it's not, still, contaminations can happen. And I saw myself this in our place. When you change out animals in the workout station, you can still have some accidental cross and contamination. But I would like today to concentrate specifically in genetic background, which is a relatively new concern. Because for many decades, like in the 70s or 80s or 90s, the only concern was the purity of the inverse strains. But with the big increase in genetically modified mice, specifically transgenic knockouts, and now with all the conditional knockout, CRE, etc., the genetic background has been more important than the original inverse strain authentication. So genetic background, basically, we can define as everything that is not everything on the genome. Of course, all the sequences on the genome that are not the gene of interest that we are working. It's not the transgene or the knockout or the targeted allele that we are working with. So all the other parts of the genome, we can consider the genetic background. So let me show very, very fast a few examples. There are hundreds of examples to explain why genetic background is really important. And I'm going to show only a few. I'm always showing this one because it's one of the first ones. You can see the publications there are actually from the early 70s, so a long time ago. And this is before genetic engineer was even there. So for many decades, again, from the beginning of mouse genetics in the early 1900s until the 80s, where we have the first transgenics, the only possibility outside outbred or inverse strains were to work with spontaneous mutations. So these are two classical spontaneous mutations that were very popular during the 60s and 70s for obesity and diabetes research. Actually, at that time, when these papers were published in the 70s, we didn't even know the genes that were affected. So they were called only obese and diabetes. Now we know that one is the leptin, it's a mutation in the leptin gene, and the other one is a mutation in the leptin receptor. So you have the pictures here, they have obesity and diabetes. But these papers are showing for the first time, early 70s, and the title is very clear, the influence of genetic background on the expression of these mutations. So this was kind of a new idea, a new concept, even though we knew that genes work in network and there are modified genes that can affect the phenotype. These were clearly the pioneer papers expressing a concern about the influence of the genetic background. You can see here that the same mutations on one background, this background is 57 black 6J, have obesity, but the diabetes is transient. However, in a different background, 57 black KS, which is a related inverse strain, you can see by the name that it's very close related, but it's not the same, it's a different inverse strain, has over diabetes. So this was the first publication, but I'm going to show you a few more out of hundreds, as I said. This is important because it's the first couple of publications. These are two publications back to back in Science. So this is in the mid 90s. And this is kind of the first publication showing the same effect of the genetic background, but on a knockout, on a genetically engineered model. This is the Egf receptor knockout. You can see here on the right, what they're showing is that the survival of the homozygous knockouts we have here on this column is very different if you change the background. So basically here what we have is the same knockout allele of the Egf receptor. This is a complete knockout, a loss of function knockout. But when you have a CF1 outbred background, you don't have any survival after birth. So the same with 129, but it's very different if we have CD1. Basically, what they were showing was that, again, I mean, how only the background is changing the phenotype. The knockout is exactly the same. The background is different. It's a different inbred or outbred, in this case, background. Another example that I would like to show always is this one, because it's very compelling example. This is from the 2000s, and this is a very popular model for cancer research. P10 is a tumor suppressor gene, like P53, for example. And we have this model that the homozygous knockout, complete constitutive knockouts, will die. However, heterozygous, so you can see here plus minus, will survive and will develop tumors. One of the tumors that they develop is lymphoma, as you can see here. But look at this, what I'm showing here is very striking. The same exact genotype here, heterozygous for P10, if they are in a mixed background with 129, which is the original background of this knockout, with BALC, they have almost no incidence of lymphoma. It's less than 10%, it's maybe 5%. But however, the same exact knockout gene has 90% lymphomas in these different backgrounds. It's a huge influence of the background on this model. So this is very important. You cannot do anything. I mean, you cannot test any drug with effects on lymphoma if you have mixed background. So that would be dramatic to do that. And I am also taking the opportunity to show how bad the nomenclature is here. I mean, maybe you're not familiar, but this is a horrible nomenclature in this paper, which is very common in all of the many publications. One last example, and only just to show you more examples. This is, again, it's a different model, because this is actually a model of dystonia, and again, how the survival is very different. If we have a hybrid F1 mice that have 129 with DBA2 component versus 129 versus V6. Again, these are just only examples. I'm not going into the details. I'm just trying to show you how important the genetic background is, because it have a dramatic effect on the phenotype in many cases. Of course, not always, but many cases. Another example, and because this is just very recent example, 2016. And again, this is actually another spontaneous mutation that is a model for this muscular dystrophia. This is the MDX model, but again, the phenotype is very different if you have 129 component or you have B6 component. So another example of the same. What, I mean, how is this problem originated, basically, this mixed background? Why we have so many mixed backgrounds? Well, this is a historical reason. I mean, since the early 1990s, that we've been this increase in the use of knockouts and knock-ins through homologous recombination and through the use of embryonic stem cells. What happened was that for many years, until very recently, the embryonic stem cells used for the development of knockouts were all of them coming from 129 family of inverse strains, 129S family or P or X1. There are different members of this family. So they were doing the in vitro work on these ES cells. They developed the chimera. Of course, I don't have time to explain now all the technology, but in 2 words, after obtaining the chimera with the ES cells that are targeted to the gene of interest, what we normally do is to cross with the wild type animal to obtain something that is called germline transmission, to be sure that the chimera is transmitting the targeted allele to the next generation. So what happened here is, if we consider that this is normally a male, the sperm cells of this male will be originally from the 129 inverse strain. This is the female, so this first generation here, this are technically or genetically speaking, hybrid F1s, because they have 1 chromosome from the father here that is completely 129, in this example X6, and from the mother, of course, the oocyte, and this is 1 chromosome each. What was the problem is that nobody wanted to work with 129 as an inbred strain because it's not a good breeder. There's not much information. Everybody was, of course, leaning to use Syphilis Black 6. That is the most popular inbred strain used for knockouts and knock-in. So what they are doing, and is still mainly doing, is just intercross these guys here that are called F1s. And we will obtain is called F2 population. And in this case, all of these animals are genetically different. I mean, they will have, on average, 50% of each of the original parental strains, but individually, these mice are different. So different pieces of chromosome will have different components. And that was the problem for many years. People is crossing the mice like this, people are crossing mice like this, and they're just expanding the colony from here, and they're using the mice that they want for the experiments. All those mice, of course, are different. They have different background because they have like a different patchwork from the original parental strains, and that's a problem. The same with transgenics. I'm talking about here about the classical DNA pronuclear microinjection, additive transgenic. That was the first technique available in the early 80s. So many laboratories that are working on transgenics like to use hybrids for the injections because they are strong embryos. That is maybe fine, but the problem is down the road, if you don't do back crosses, you may end up with this type of situation where you have, for example, on the top panel, you have 3 transgenic mice that are PCR positive for your transgene. But since these are coming from hybrid crosses, then if these are F2s or F3 or F4s, I mean, down the road, you have all 3 positive here. Down here, you have 3 that are wild type or negative to the PCR. But individually, each mice is different. I mean, because they have different colors, as you can see. I'm showing here 4 sets of chromosomes, random chromosomes. And the colors, of course, will represent the genome of the original inbred strains, parental inbred strain. So we are working with mice that are basically transgenic positive or wild type. Yes, that's nice, but we are working with mice that are not genetically identical. So that could be, could be potentially a problem. What is doing a lot of problems now, is of course that we are using a lot of conditional models, and all the conditional models also the same for the also the doxycycline also models that are inducible models. All of those models require to cross 2 different inbred strains or 2 different lines. One line is the one that we have the gene of interest, that is flux by these LOXP sites. And the other line that is normally available through different places, some commercial or some simple just repositories or collaborators. So normally the more typical situation is that these we call Cree-expressing lines are not always on the same background of the LOXP, which is a very common situation. Then we need to cross, and there's no other way, because we need to have the Cree and the LOXP on the same mouse, like here. I borrowed this from the Jackson laboratory, this slide. When we have this is the problem. If this Cree line is different background from the flux or LOXP line, then we have again a mixed background, and we normally start to intercross these mice to generate the different Cree positive, Cree negative, homozygous, LOXP, etc., etc. So all those are again a mixed background that could be a problem. This is a very nice paper from 2016 where they decided to do a very elegant design of experiment to show how really genetic background has an influence on different knockout. Look at what they did here. It's very, very nice. They decided to cross different knockout mice, in this case, loss of function alleles, complete knockouts, on a pure B6 background. They cross heterozygous males like this with a set of females coming, all of these individual females, you can see the colors here, from 30 different inbred strains. That's why all these colors. When you cross, of course, two lines like this, what you generate is technically called hybrid F1. Again, one chromosome coming from the male, one chromosome coming from the female. But what they did is to generate 30 different hybrid F1s, where, take a look at this, these are heterozygous. So what you generate is 50% will be heterozygous for that knockout. The other 50% will be wild type. But every combination will do, of course, 30 different combinations of hybrid F1s, basically. And then what they did, they choose a series of knockouts. I'm showing only one here, or a couple, or actually a couple of genes. They have a list of more genes. You get the reference, you can read the paper if you're interested. So they pick up different genes and they study the effects. In this case, the phenotype is an acoustic startle response. I'm not familiar with what the genes are doing, but what I'm trying to show is how the different inverse hybrid F1 behave. And what is striking from this paper is the opposite effects that are coming from the background. What I'm showing here is how the wild types is the black is wild type here. How the wild type behave in one extreme, I mean, with higher response than the knockout, but on the other extreme is exact opposite. The wild type has less response than the knockout. Which is producing this, or what is producing this? Only the background. This is the exact same knockout. The chromosomes are exactly identical. Remember, hybrid F1s are genetically identical because they are getting one chromosome from one line and the other chromosome with the other. So with the only effect here is the background. And this is the same exact type of situation. Another gene and two complete extreme results only because of the background. And these are statistically significant. You can see here the asterisk showing that significance. I'm slowly moving to something that is also very important today, and it's all related, of course, with the same situation of genetic background, but it has to do more with the substrain of the particular background, because again, it's something that investigators are not paying attention. I mean, at least for me, striking lack of information about substrains. Many researchers or students or postdocs completely ignore the existence of substrains. And also, the substrains could be dramatically different in some cases, influencing the phenotype. Not only different inverse strain, also different substrains could be important. Basically, I'm going to move here fast, but basically what is important is the last bullet here, is that when we split colonies and we send animals to other places, after 20 generations of separation, actually this is only 10 generations of each place, I mean, we can consider this as substrains. And this is something that happens all the time. I'm going to show very quickly only two examples, and I'm going to concentrate specifically on the syphilis and black cyst history because it's a more common situation. So BARC, for example, is a very popular strain used specifically in immunology. So many of the immunologists use BARC. However, we have several substrains, but there are three that are very common: the Jackson substrain, the Underwood NIH substrain, and the Bailey-Jackson. As you can see here, down here, I'm not trying to do any advertising, but this is something that always strikes me, because normally many of the users or the buyers of this substrain don't know there are different substrains out there. And they believe that when they go some days to one place and to purchase mice, and they maybe another week or many months down the road, another student decides to go to another place to purchase the mouse. They're purchasing different mice. This is not the same. BARC-J is not the same as BARC-AN NIH. N is the NIH, is the laboratory code for NIH. J is the laboratory code for the Jackson. So this could be really different. And I'm putting down here, actually they are very different if you are looking into doing production of antibodies, because they will respond very different to the injection of this antibody production. And this is a paper that shows very clearly this, because they are comparing, in this case, two substrains that are actually close. I mean, closer than the others are showing. This is the Bailey versus the Jackson substrain. So this heat map is showing the genes that are, turn on or turn off. And as you can see, they're very different. This is a model of arthritis that is produced by injection of this proteoglycan substance. But what I'm only trying to show you is how different they behave. I mean, before and after the injection of this compound. So you can see by the colors how they behave really different. If you use the Bailey versus you use the J substrate, they are different. I mean, they have different percentage of course of arthritis scores, etc., etc. I apologize, I'm moving fast, but my idea is only to show examples and to convince you that this is very important. Then you can go yourself and read these papers. Also, they have differences in behavior. And the main problem, of course, is this one, because it's the most popular inverse strain out there for knockout, knock-ins, and anything that is genetically modified, is that the number of substrains that the C57 black 6 has. And this tree is showing how complicated is this. Basically, we can concentrate only on two groups, two substrains. One is the NIH, which is the N here. Why is that? Because from the original substrain that was developed at the Jackson at the time in the 20s, in the 50s, there was one group of mice that was sent to NIH, and that originated all of the ones that we know now under N/N, because all of those belong to the N substrain. And then, of course, we have several of the different substrains that are most of them in Europe, that are also different to the NIH. So we have the J substrain that is coming from the original Jackson of the 1920s, that of course it should be different to the one that we have now, because of all of those years, almost 100 years of genetic drift. Let me show you a couple of details about these two substrains. Again, I mean, what I'm trying to show here with this picture is not to advertise, but to show you that if you go to the web page looking into B6 mice, and you're purchasing these mice, you need to be very aware they are two complete different products. You have the J here, has one number, stock number, but the N, you need to pay attention to the N here. The NIH, it has a different catalog number. This is a different product. It's not the same substrain. The J at the end means only that they are produced and sold by the Jackson, but you need to pay attention to the letter after the 6. So they are two very different. Of course, they are very close related. They are more than 99% identical, but that 1% they are different could be important, and I'm going to show you how. A few years ago, 2013, for the first time, they sequenced completely both substrains. Remember that the original mouse genome sequence from 2002 is a C57 black 6J female. But now, I mean, since 2013, we have the complete sequence of the N substrain. So if you like to take a look at this paper, all of the information is there. Let me go into details of some genes. I mean, when we have some genes that are affecting the phenotype, this is part of the same problem. It's part of the background or part of the substrain issue. But some papers like to call these passenger mutations, because they are they are present on these substrains or different strains, and those are the mutations that are producing a problem or a change in the phenotype. It's just a technical term. So there is one that is very, it was known for many years. And this is, in this case, we're talking about the C3H strain, another classical inverse strain from the 1920s. However, and same as the case for the C57 black 6, They have one that is the one that was maintained at the Jackson laboratory. That's why it has the J as a laboratory code. But the other one, again, was also sent to NIH, and that started another substrain that is called the N. Well, these two substrains have one particular gene that we could call passenger mutation in this case, on this TLR4 gene, that will produce a complete different response to the injection of this LPS from bacteria endotoxin in that injection. So the response will be completely different, either if you use the J or the N, because of this particular mutation on the defective allele of this gene. So actually, this is a Nobel Prize that is in Dallas, close to us in Houston, at MD Anderson. Another example that it was actually affecting our department in Smithfield at MD Anderson, because we were doing at the time a lot of skin cancer research, was this that was published in 2008. And it is, again, a passenger mutation on the FBB, on one of the FBB substrains. And this is, again, this is FBB only from the taconic production colony. Will have a mutation on this gene that is called SKIN1. Again, I'm not presenting any details, but you can read from the title that they have, this gene has some function on the gamma delta T cell response. And since this has a lot of effect on the skin, and this gene is technically, I'm showing that how it's a truncation mutation, so the protein will lose all of that part. So basically, it's a knockout. It's a functional, spontaneous knockout of this gene that is not producing an obvious phenotype externally, but will have an influence on the response of the gamma delta in the skin. So what happened here is that we had to genotype hundreds and hundreds of mice at our department, because, again, as I was presented at the beginning, people normally don't know what substance they are working with. When I ask many researchers where the FBB were coming from, most of them had no idea. That's the reality. There was a word, no, I don't remember. Sometimes we purchase from Taconic, sometimes we purchase from Trans River, etc. So, and that's again a problem, because they are not the same. I mean, in this case, this passenger mutation is only affecting the taconic animals and not the other FBBs. Again, these are only examples. That's why I'm just moving fast through examples. Let me spend a little more time on the citizen black group of substrains. So again, what happened with the J or the Jackson substrain is that in this paper is showing that in 2006, they discovered a big deletion on this NNT gene that is only present on a group of Jackson substrain. As I'm showing here, down here, this same deletion is not present in all of the other substrains, neither the NIH substrains, but neither in the other of the all different citizen black strains, even the 10 or the J or the 58, etc. These are all related substrains. They are not that common now. I mean, they are not that popular. But which is really popular is the N, of course. So the N has a wild type allele of this gene, and the J has the mutation. That is a deletion, as I said, but basically, again, is a knockout. It's a functional knockout for this gene, spontaneous mutation. And this is a paper that's showing this very nicely. That's why I always use this paper as an example, how this particular mutation will affect the phenotype in some cases. This is a group that was working on the JNK2 knockout model. With these animals they are okay, they are alive, but they have some impact on the liver. So what this group was trying to do, knowing about this mutation, they used two different groups as wild type controls. For wild type for the JNK2, but they, knowing that, they say, okay, let's use B6 from the Jackson as a wild-type control for the JNK2, and there's also, let's do the same experiment with the NIH as control. But however, as I'm presenting you now, the Jackson has this knockout, that is spontaneous mutation on the gene, and the NIH is wild-type. Which is really striking, that's why I present this paper, is they obtain a complete opposite result if we use one substrain as the control or the other substrain. So in one case, this is ALT, is the hepatic enzyme that they're measuring in this case. But if you only use this as a control, you could conclude that your gene JNK2 has a hepatoprotective effect, because the levels of this enzyme is lower. When the liver is affected, normally these enzymes are higher. On the other side, as you can see, the Jackson the NIH substrain is giving the opposite result. So you could conclude that this particular gene has hepatotoxic effect. Again, I'm not going into the details into the paper. I'm only showing you how important it is to know if we are using Jackson substrain or you're using NIH. That's what I'm saying. A very similar example now is from the other substrain, the NIH substrain. The N substrain has this mutation, again, spontaneous mutation, passenger mutation, on the CRB gene that is producing a defect on the retina. So it's a retinal degeneration 8 is the name of the mutation. Now we know the gene, but basically these mice have a phenotype on the retina, and this is also affecting, and this is very important, all the ES cells that are produced using the NIH substrain, which is something very common from some of these UCOM consortiums. Like all of them use the NIH substrain. And this also was something that affected in our institute, because there was a researcher working on a knockout that has I phenotype. I was completely confounding results because he was using a mixed background of J substrain and NIH substrain, and there was a really nightmare for this researcher. We had to do a lot of genotyping and clean the colony, only because there was not information about the mutation on the NIH substrain. So the situation is really more complicated. I'm not going again into details, but there are other mutations that are out there in other substrains that are more popular in Europe than in the US. That this OLA substrain has this big deletion affected to genes, for example. And of course, you have all the combination of different substrains that will affect different genes. I already mentioned the NNT here and the RD8 here. And again, here, a very recent paper, if you want to read more, this is a last year paper, you can read it. They have all the information. Again, the NNT mutation, the C-nucleate deletion, another deletion on the Y chromosome here, only on this group of taconic mice, and the RD8, and another one on the DOC2. So this is really more complicated than we knew. And of course, we're knowing all of this big, again here, all of these mutations we are knowing because of the sequencing. Now is so cheap and available. We are sequencing a lot of strains. This is actually something that we normally call quiet mutations, and we also in our lab we discovered one a few years ago that several inverse strains also have this mutation on this gene that is called GPR84. These are quiet because they don't show a striking phenotype on the behavior or on the skin or the color or something that we can see. That's why they are called quiet mutations. So we are knowing now, as I said, because we have all of these inbred and wild mutations that are now completely sequenced, and you can have the information from the ensemble or the different places that now we know all these, and that's why we are knowing that all these passenger mutations are there. So what are the solutions? Basically, the more important is, of course, to be informed and to know that this is a problem and we need to read about it. And to use, of course, try to use pure inverse strains when you are doing genetic manipulation, or even CRISPR-Cas9 that is so popular now. If you are still doing the old way, chimeras or ESLs, of course, try to keep all the system in the same inverse strain. Use the same inverse ESLs as the inverse strain of your interest. So try to avoid all of these mixed background. But if you already have your line for many years, or you're still working with CRE-logs, conditional system, of course, the solution is to do background characterization. At least you need to know what you have. I mean, it's not good to ignore what you have. And of course, after the background characterization, the speak congenic or the congenic development will be the more, the recommendation will be to develop congenic strain, which is something that is very easy to do, but it takes, we need to use genetic markers, of course, either to do background characterization or to do speak congenic or marker-assisted congenic. You need to use genetic markers. Typically, we use microsatellites for many decades, but now we are moving almost 100% to SNPs, because this is a more common genetic marker today, and there are millions of SNPs in the mouse, in the order of 50 million, so we can pick up the different markers that we want to use for the different combination of inverse strains. So microsatellites are still used, but really now SNPs is the marker of choice. For background characterization, what we do in our service core normally is this. We use 100, 120, 150, around those numbers to do the background characterizations, and we try to estimate, of course, the percentage of alleles that are in the different animals that we are studying, in order to tell the investigator what is the background of their line. So this is an example of background characterization from our service at MD Anderson. This is using SNPs, as you can see on this column. We have all the chromosomes. Remember, the mouse has 19 autosomal chromosomes. We are, of course, placing SNPs on the different chromosomes, trying to place them in a way that we cover the whole genome in a way that we don't leave gaps. It's not really needed to do 500 SNPs or 1,000 SNPs. I mean, it's more than enough to do 100, 120, or 150 SNPs if they are evenly distributed. And then after doing this analysis, of course, these markers are normally polymorphic, so they are different between the strains we are trying to study. And this is a real case of background characterization. I mean, you can see, well, with these animals were only 80% or less, 79%. 60% black 6, which is the background that they want. And we normally place the genes of interest. This was a very complex line, which is something very common today, with different genes. You see, with different targeted genes. 4 different genes in the same line is being common now. And it's part of the problem, because as you can see, there's a lot of 129 areas on these animals, some heterozygous too. And we are trying to clean this and to do more speak congenic or more back crosses onto B6, which is the background of interest for this investigator. So this is what we do. How we do speak congenic, I'm gonna move fast because we don't have much more time. But remember, this congenic development is based on back crosses. And back crosses is when you use your gene of interest, in this case is this red dot here. Of course, you are trying to move from the donor strain to the background strain. Normally B6 is the more common background here. So when you go back to the original strain, that's why it's called back cross. You do this for several generations. So you have on the left the different percentages of background, how they're increasing with time. And each round of backcross, we use the letter N. That's the nomenclature that we use for the number of backcross generations, of the N2, N3, N5, etc. So typically, before the use of genetic markers, we consider that doing 10 backcross generations was okay, and we were getting around 99% of the background of your interest. That would be after 10 generations, and that's of course selecting the gene of interest at each time. But today what we do, and that's what is called marker-assisted backcrossing, and it's based of course on the use of SNPs, is that we have now the possibility to choose which particular N2 or N3 or N4 animal has the more background that we want. In this case, again, 4 different chromosomes, random chromosomes. The green is the background that we want, the color that we want. Of course, if we can choose this mouse instead of the other 2 randomly, then that means that this is our best breeder. And then we speed up this process. Instead of 10 generations, we can do only 5 or even 4. And that, of course, is a big, huge saving in money in the animal facility and time for writing grants and doing experiments. So we can cut the time in maybe half. So the expense of doing the background characterization is really much better than all the years of backcrossing. Okay, so this is what we normally do. We do the crosses. I mean, the PI is doing the crosses. We only do the background characterization. So we do this at the basically starting at the N2 only, because the F1 there's no need to do background characterization of the F1s. We do that on the N2s. We use this 100, 120, 150 panel, and we select the best breeder, and then we, of course, the PI keeps doing the crosses, and then we do the same at the N3, and then we do the same at the N4, and we basically doing that is we normally at N4 we are normally at the 99% background level, so that's what we want. And I think with that, only one more slide is to show that still after all of those back crosses, we need to keep in mind or consider that the area that is surrounded the gene of interest, that is called the flanking gene problem, even though we move up until generation 20, still, of course, we'll have some areas flanking that are from the original background, because, of course, that's the linkage effect. It's very hard to get rid of that. But eventually, we could use more markers on this area to select the best breeders that have less of the flanking genes from the original donor strain. I think with that, I'm concluding. Again, what is important here is to know your strain and your substrain. I mean, be sure that is the substrain that fits your research. And then from that, of course, use good nomenclature, as I said before. Be very vigilant about the background. I mean, it's very important to have a pure background and to know which is the background that you are using. Don't change background in the middle, of course, of experiments. And again, number one is to do the background characterization. I mean, you cannot do an experiment blindly without knowing what you have in the background. So develop the congenic strain, either classical or speak congenic, much better. All of them actually has to do with also with the refinement and reduction. So it's important from the ethical point of view that we are not wasting animal lives, and we're using the best model that we can, and that's part of the refinement and the reduction. Two of the R's that we are also talking about. And with that, I will be happy to take questions.
Well, thank you so much, Fernando. While we give people some time to type in any questions they might have, go ahead and use that question section on the right-hand side of your screen there, and we'll get those answered for you. I'd like to mention, sorry, getting set so I can control the slides, that Transgenetics is offering now, in addition to our standard genotyping service for tracking that gene of interest, we are now offering a genetic monitoring service. We currently offer two different use cases or two different panels here. We offer a full strain in-brain strain testing of the C57 Black 6 versus 129. And then we also offer a substrain panel looking at the Black 6J versus Black 6N that Fernando spoke so much about. We'll also be adding a BALB-C panel versus Black 6 very soon. Look for that in a month or so. This service is available on all samples that were genotyped here at Transgenetics, so you don't need to collect another sample from your animals, which again reduces the handling and stress on those animals. Results are returned within a week, and those full strain panels are at a price of $150 a sample. The substrain is $60 a sample. So with that, we're going to start taking some questions, or excuse me, answering some of your questions, and we'll go forward from that. What I'd like to do is remind folks also that there will be a recording of this webinar available, and once that is available, we'll send out the link. So we did get some questions about the availability of the slides or this information, and that will be available to everyone, and we'll let you know as soon as it is ready. So first question, how many offspring should be tested per generation for a speed congenics problem or a marker-assisted breeding project?
Okay, that's a very good question that I have all the time. In the original papers that they describe and they actually gave the name speed congenics, this paper from the 1990s, they recommend to use 20 mice. Of course, they need to be carriers of the knockout allele or the transgenic alleles. They recommended to use at least 20 or around 20 mice for that. However, after more than 10 years doing this, I could really tell that that's almost never the case. I mean, we've been doing hundreds of speed congenic projects in our facility, and it's very hard to get that high number, really. We normally are okay. We recommend to at least a minimum of 8, ideally 10 or 12. But we've been having good results even with 8, and we have a lot of data I can share if someone wants to share to see that data. But we've been, of course, this depends with luck sometimes. This is because these are random recombinations. This is a little bit like a lotto. I mean, if you get that mouse that is a perfect mouse, I mean, you can maybe do it in 9 months. I mean, but of course, that all depends on luck. But we're having normally, I would say, like from a year to a year and a half, using 8 mice for the marker-assisted characterization to have at least 99% of the desired background.
Great, thank you. Another question, is it possible to inadvertently create a substrain in the lab? If I breed my own C57 Black 6 and have continued for over 10 years, should I be concerned?
Well, yes, of course, the answer is yes. I mean, the recommendation actually is not to do that. I mean, it's to repopulate your colony from the vendor of your choice every 3 to 5 years. I mean, at least. So if you choose your substrain, choose your vendor that is better for your experiment, and refresh the colony from those vendors, because those vendors are doing cryopreservation. They're keeping, of course, a genetic monitoring program that is good. Normally, that's my recommendation, because yes, you can create a substrain and you can inadvertently have a passenger mutation that you don't know, unless you want to, of course, do exome sequencing and describe your particular substrain. I am getting a lot of these questions when I travel around the world, and specifically in South America. where they really are keeping substrains for decades. I mean, in those cases, if they don't want to change that, they need to rename the substrain. They need to find a new laboratory code, and they need to put on the papers that they are using a particular substrain of the inverse strain, original inverse strain.
Great. Then how do we use this information to aid in the design of our experiments to make sure that they have the most reproducible results and results that will most accurately reflect translation to human clinical applications? So I think the question there is boils down a bit to maybe there's some bullet points or highlights of what would best practice kind of be when you're setting up an experiment.
Okay, yeah, I think, well, yeah, not only setting out the experiment, but what is important is also when we publish, I always recommend include specifically in materials and methods, which particular substrain, and ideally is actually to put the vendor and to put the stock number in the case of the Jackson, or the catalog number in other cases that where the mice that you are using that particular paper are coming from. If you have a genetically modified mice that is a complex background, a mixed background, again, well, if you have background characterization information, it should be very important to include that in the materials and method, that a background characterization using 150 SNPs was done with a result of 99% B6 background. You need to include that on the publication in order for other laboratories to be able to replicate that experiment. Yes, they need to know that.
Great. Well, we actually have one question here that I think I'll answer because it's regarding submitting samples for genetic monitoring here at Transgenetics. You don't actually have to submit samples. We'll already have them here, so you can go ahead and order the samples that you've already sent here for genotyping, and through your account, and order the background test or genetic monitoring test that you're interested in. Thank you so much for joining us today. I'd like to thank Fernando very much for taking the time to talk about this very interesting, slightly scary topic.
Thank you for having me.